Hey there. How's everybody doing?
Good with Wi-Fi.
Hey, so you're back.
Back.
All right, good. Well, the college has evidently decided to rip out the stairs that come up to White Science Center from the faculty lot and the stairs that come up to White Science Center from the quad. And so you may hear very loud jackhammering noises during today's episode of Animal Behavior Remote. So just bear with that. Not a lot that I can do about it. It's been going on all morning. So anyway, everybody seems to be hanging in. All right, then we'll begin. We were talking about mating systems before, and we had just begun talking about how you go about assessing polygyny or polygamy, either one, but basically how you go about paternity testing. I mentioned the use of microsatellites. One of the nice things about microsatellites is that they're relatively cheap to process, and they can reveal patterns of fertilization. In this case, the example in your book is a situation with red-winged blackbirds, where you can see who's achieving extra pair fertilizations and with whom they are achieving them. And you'll notice that there is a spatial pattern to this. Typically, individuals are mating with other individuals outside their own territory in territories that are adjacent to them. Microsatellites are, I think, probably more commonly used nowadays, but back before microsatellites were well developed, there was restriction fragment length polymorphisms. Once again, they give you similar kinds of data. In this case, you have one microsatellite locus. One of the advantages of RFLPs is that you can simultaneously assess lots of loci. So you guys remember what restriction enzymes are? Who can tell me what restriction enzymes are? Oh, I'm not showing my screen, am I? I'm not. So here is the microsatellite locus, and here is a restriction fragment length polymorphism gel. Who can tell me what a restriction enzyme is? Anybody? You should remember this from genetics. I imagine, well, I know that I've talked about it in genetics, and I'm pretty sure Dr. Reynolds has.
Doesn't it cut DNA up?
Yes, that is exactly what restriction enzymes do. So you will have a DNA molecule, and the DNA molecule is a long linear molecule, because that's the way DNA is, as you know. And restriction enzymes cut the DNA at particular recognition sites. And so each restriction enzyme will have a particular set of usually 6 or 7 base pairs, and it will recognize that restriction site, and it will cut the DNA at those sites. And so each of these cutting sites is a restriction fragment, a restriction enzyme locus. And so when you cut this DNA up, it results in a kind of long fragment, a slightly shorter fragment, a longer fragment, and in this case, a really long fragment, fragment 1, fragment 2, fragment 3, and fragment 4. So if you possess restriction sites at these 3 places, you'll produce 4 fragments of these links. If you have a mutation such that you are missing one of these things, you will basically have 3 fragments, and these middle 2 fragments will be replaced by a longer fragment that is the exact length of the 2 other fragments. And so the size of the fragments then tell you indirectly where these restriction sites are located, and then you can run these fragments out on a gel. One of the nice things about restriction fragments with linked polymorphisms is that the restriction sites themselves occur all throughout the genome. And so with a single restriction fragment, or with a single restriction enzyme rather, you can basically survey hundreds of different cutting sites with a single enzyme. And so it gives you many more markers than, for example, microsatellites do. Microsatellites, you have to assay each locus separately, separate primers for all of these loci, etc. And so that's a little more more time-consuming, although it gives you basically the same kind of information. This is just one of the cases where, in the case of an older technology, it actually gives you more information than a newer technology. So if you're doing this for, once again, humans, which is where RFLPs are used a lot, you have a mom, you have a child, and you have 2 prospective parents. You see that these males share, both males share this marker with child, so that particular restriction site isn't particularly informative. And what you see is that male 1 has contributed a marker here, male 1 has contributed a fragment here, and a fragment here, and a fragment here. And as a result, it's fairly obvious that this child belongs to male 1, not male 2 in this case. And all paternity testing takes on this type of marker-based analysis. It just depends on what kind of marker you're looking at. Questions about paternity testing?
Do you expect the child to share all the markers that the male does, or roughly half, or what?
It will depend, but any marker that isn't found in the male and in the child should be found in the female. So this is a marker that the mother doesn't have, the child doesn't have, and the father doesn't have. So the child inherited the lack of that marker from this male. And so where you see a lack of marker both in the child and the father, that's also representative of, you know, being the child of male 1, not male 2. So if you look at every marker that the child possesses, they have to have either come from male 1 or the mother. Male 1, mother. Male 1, mother. Male 1 and mother, it looks like. Male 1 and the mother. Male 1 and the mother, and looks like just the mother. So every marker that you see in the child should be found in one or the other of the parents, but not all of the markers of the parents will be in the child because they only get half of the parent's genome. And you can be heterozygous for restriction sites, just like you can be heterozygous for any genetic marker.
So are these markers measuring the length of the cut pieces or the actual location of the site?
They're measuring the length of the cut pieces, but the length of the cut pieces tells you where the locations are. So if you have a deletion that inactivates this, a deletion or an addition that deactivates this restriction site, this small fragment and this medium fragment become one large fragment. You then possess that large fragment. That large fragment stays at the top of the gel rather than traveling through the gel faster. And that fragment becomes a marker, but it's a marker because it's telling you what's the length of the DNA between either restriction site A and C, or restriction site A and B and B and C. Does that make sense?
Yeah, that makes sense. So, like, are you saying that every male will have and female, I guess, for that matter, has a different set of markers? Or like, what's the likelihood that there's enough mutations to be able to tell one male apart from another?
Oh, it's really pretty easy. So in this case, you have a mother who has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, or 15 different fragment lengths from this particular restriction enzyme. Then you have a father who has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 fragment lengths. So one of the things that you might expect, for example, this fragment length here that is found in the child may actually be due to the fact that this individual has site B functional in him. The mother doesn't, and this fragment here is basically this fragment here that's been broken up in two. And I realize that this occurs on a log scale and all that, but that's what will happen is this one doesn't appear at all because this is normally a site that would have a restriction, or that would normally not have a restriction site in it. And this one does have an additional one that has basically broken the DNA up into two smaller fragments rather than one big fragment. So typically what you do if you're doing this in a lab where you do this for a living, and this was one of my more eye-opening experiences in my scientific training. I actually worked at a lab that did paternity testing when I had just gotten out of graduate school. And I got out of graduate school, didn't get a job because I went to Peru to help my then wife at the time, to help her with her dissertation research. And we were in Peru for 3.5 months, and then we came back. It was Christmas time. I went on the job market, and I actually got a job at an optometrist's office for a while. Hated that job because I didn't like the guy that I worked for very much. And while I was bemoaning my plight, a friend of mine said, "Oh, there's this genetic testing lab over in Dallas that is hiring. You should give them a call." So I gave them a call, went over there and filled out an application and had a job interview with the owner of the company. And the owner of the company was like, "You're an ecologist and we're a genetic testing lab. And like we normally hire people with like master's degrees in biotechnology." I'm like, "Okay, that's fine. That's a well taken point. But what I do have is I have a PhD, and you know what a PhD means? It basically means you're infinitely trainable." And so I said, "I'll learn whatever I need to learn to do this job, and I'll learn it faster than any of the other people that you've hired. And if I don't, just fire me at the end of that time. I mean, what do you have to lose, really? You know, a couple of days of training. You're going to have to train somebody anyway." And so I went through and I got trained on everything that I needed to learn to do it, and I kept my job. What shocked me was what they did was they handled cases, court cases where people were getting divorced, mothers wanted child support for their children, and dudes, because dudes are tools, were basically saying, "Why should I pay you child support? Those children are not mine." To the rate of 600 cases a day came through the lab. And there were 3 teams, and each team would work 3 days on this. It took 3 days to run the process through totally. And every 3 days, a team got a new set of cases, and they averaged around 600 cases a day. So one thing I was shocked at was just the sheer volume of business that this company did. We used typically for every case, we would use 3 different restriction enzymes. So we had 3 different sources of markers, essentially. Each of these markers would give you, 15 to 20 different fragments on each gel. And then, so we had a lot of fragments, 45 to 60 fragments that we would then compare. And so once you have that many markers, it becomes pretty clear when there's a consensus and when there isn't. And so using multiple markers that have good coverage across the genome gives you a pretty good idea of the right answer. So I was shocked at the number of cases. I was pleasantly surprised at the number of people who are not seeking out extra pair fertilizations, because in most cases, the male that the female said was the dad was the dad. And at the time, to get this tested, the retail price for lawyers and stuff to do this at our company, at the company that I worked for, was about $1,600. And so inevitably, if the dude was the one who was calling for the paternity test and he lost the case and had to end up paying child support, he was also going to have to end up paying all of the court costs associated with that as well, including the $1,600 paternity test. So it was a really losing proposition in most cases for the father. And then there were just a couple of cases where it was just catastrophically bad. There was one case that I remember where there was a mom, and I think she had 5 children, and she had 3 prospective fathers, and none of the fathers matched any of the children, such that we went back and reran the entire case again and basically got the same results twice. We wondered whether we had screwed something up, and it just shocked me what that person's life must be like. So it works pretty well, actually. It works well enough that it holds up in court cases.
Do you have to have information from 2 males, or if you just had 1 male, could you like kind of?
No, so you can have information from 1 male. If that male then isn't the parent, then most of his markers will not show up in any of the children, and you can rule that male out as the father of those children. So it works just as well to exclude a male as the father as it does to include a male as the father.
Okay.
Now, there are cases, and we didn't see any of these while I was there, but there are also cases where people have been involved in these kinds of things, and they discover that the child is not the child of the mother. And in those cases, it's because the child has been abducted. And then that sets in a whole other set of issues on the human side of things. So you can do this in humans, you can do this in mongoose. It doesn't matter. Everybody's DNA is the same from this perspective. Other questions about paternity testing? All right. The polygyny threshold model. The polygyny threshold model basically is a model that attempts to determine why you would enter into polygyny. So in some cases, females choose polygyny because there's not much to be gained from male parental care. So when you enter into a polygynous relationship, one of the things that you can probably count on is a reduction in parental care from the male. And so there is a cost to polygyny in that you're sharing resources with whoever else is in a polygynous relationship with the male, even if the male is not providing any parental care. Presumably the male is maintaining a territory that has resources in it. So the way this works is when a female joins a male, the female can either join a male who already has another female, or a female can join an unpaired male. If the female joins the male in circle A up here, she's entering into polygyny. If the female joins this male in B, she's entering into monogamy. And presumably, if you're in a monogamous relationship, you're just going to have more resources available to you. And so the monogamy curve here is going to be higher than the polygyny curve. Being the first female in a monogamous relationship is always going to be better than being the second female in a polygynous situation. And so the difference between these two lines is the cost of polygyny. Now, these curves vary on the basis of territory quality. Territory B has low territory, territory A has high territory. And this A and B in territory quality applies to A and B in these circles. So this female has to decide, does she join territory A up here, where there's already another female, or does she go to a territory here? Sorry, does she join as the second female, territory A, where this male is, or does she become the first female in territory B with this male? And it's going to all depend upon what the shape of this curve is and how far apart this cost is. In this case, joining territory A as the second female gives you higher reproductive success than joining territory B as the first female because of the shape of this curve. And so you have to be beyond this polygyny threshold in terms of territory quality in order for you to become polygynous. And so in this case, it's much better to be the second female on territory A than to be the first female on territory B. So what's going to matter in terms of determining this decision, do I enter into a polygynous relationship or do I go for a monogamous relationship, is the nature of these relationships between territory quality and female reproductive success in a monogamous relationship versus a polygynous relationship. So here we have two scenarios. In the first scenario here, there's a big gap in territory quality, such that the first female on site would want to go into territory A and be a monogamous female. In territory A and be a monogamous female. Territory B is similar in quality to territory A, and so it's better for the second female to not enter into polygyny there, but to pair with the male in territory B. But then there's a steep fall off in territory quality between A and B and C, D, E, and F. In which case, female 3 and 4 should enter into polygynous relationships, become the second female on territory A and B. But once that is done, then individuals 5 and 6 become monogamous individuals in territory C and D. In the other situation where territory quality is quite high and all the territories are very similar, it's much better to, once again, to be in monogamous relationships and get the full benefits of that high territory quality. And only after you've gone down the territory quality scale somewhat does it become productive now to be the second female in these highest quality territories. So the specifics of territory quality, how it relates to female reproductive success, determines what the decision is that gets made by females in choosing whether to join an already mated male or not.
So
yeah, go ahead.
the people or the numbers, the organisms on the pink line aren't together? Like, I'm confused about the way it's drawn.
These are single females. So this female is in a monogamous. So the pink line is the monogamy line. The blue line is the polygyny line. So
Okay.
individual one, of course, would join the male in territory A because that's the best territory. But now he's got a female in territory A. So female 2 has a choice of joining the male in territory B, who's unpaired, or becoming the second female in territory A. Well, it's a much better deal to be the first female in territory B than the second female in territory A. Third female that comes in territory A and B are occupied. So you can be the first female in territory 3, or you can be the second female in territory A. Once again, but because there's this cost of polygyny, you choose monogamy in territory C. Fourth individual comes along, I can be the first female in territory D, or the second female in territory A. Now the gain in fitness begins to fall off somewhat, such that to be the first female in E, and we would have to draw straight lines on the graph here, presumably that's lower than being the second female in territory A. And like I said, you would have to draw vertical lines and see where they cross these lines. So in this way, it's kind of like the ideal free distribution in a sense. You join a queue until it's no longer productive to join a queue, and or you go to, well, it's like the ideal free distribution that you have these two lines that represent fitness benefits, and you join those lines depending on what the benefit is. And so whenever you have a steep fall off, it very quickly becomes more profitable to go to lower quality. It very quickly becomes better to be the second female in a territory than the first female in the territory, because these are really high quality territories and these are all really low quality territories. In this case, all the territories are relatively high quality and all of the territories are relatively similar in quality. So it's much better to join unmated males rather than to join already mated males, is the take home point.
Right. I guess I'm just a little. I think it's just the spacing that's throwing me off, because the lines look the same, but then the communication is, you know what I mean?
Yeah, what I haven't done is drawn the lines. So this is a figure from your book, but you could come in here and basically make a vertical line and then superimpose that vertical line. So D has filled up, and now E is filling up. So now that E is there, you could, so this is how you should work this on a test in reality, if you're trying to figure this out. And most of you figured out the ideal free distribution thing. So then the question is, oh yeah, and they've drawn the figure incorrectly in your book. Because what you would do is you would turn this 90 degrees on its side, and you would put it where this intersects with that. That shows you what fitness is if you become a monogamous female in this territory versus I should have just done the horizontal one again. Versus being the second female here. Yeah, so your figure in the book is actually wrong.
Okay.
Because it makes more sense for individual 5 to be monogamous here than to be polygynous here. Now, let's look at individual 6. So now there's let's see. This is why scientists should be more involved in creating the figures for their textbooks. I need a text box. So now, really, in reality, individual 5 has taken residence in territory E. So now the question for individual 6 is, oh no, so I had that wrong. I put the first line in the wrong place. So the individual 5 has to make the choice. Do I become polygynous here, or do I stay monogamous here? Where that line crosses that line is higher than where this line crosses this line, so you should be monogamous there. So now the question for individual 6 is, do I become monogamous in F, or do I become polygynous in A? And at that point, I would have to blow it up, but it looks like it's kind of a toss-up. So I would probably redraw this such that 5 would be here and 6 would be here, now that I've made myself some nice 90-degree lines. Does that make sense?
Yes.
Yeah, that's why when I give you these figures, I draw the lines on them for a test, so you don't have to draw the lines yourself, because I can create these in a computer package where I can make sure that the lines are actually perpendicular to the axes, and it makes making the decision a little better. And if I was doing this for a test, I would also alter it such that the pink line was either lower or the blue line was higher. I would reduce the cost of polygyny so that the cutoff is a little more unambiguous than it currently is. Because as I look at that, that's even hard for me to make a decision as to whether or not individual 6 should be monogamous in territory F, or if individual 6 should be polygynous in territory A. It's just too close to call visually. Actually, I think it should be individual 6 should be monogamous in territory F. So hopefully that clarifies that.
Definitely does.
Yeah. So here's one of the problems with textbook writers. Seldom do textbook writers have very much control over the like these nuts and bolts kinds of specifics of the figures. And this is one place where the author should have scrutinized this a little more tightly and said, that's not really quite right, because it's not when you start looking at it carefully. All right, so whether the figure is made correctly, this is how you figure out whether you should enter into polygyny or not. So
what about like polygyny where you're not necessarily like taking up a territory with a male? You know, like what about females that just kind of like roll in and the male mates with a bunch of females that roll in and roll out?
Well, and in that case, you're not gaining any resources from the male. So in that case, territory quality doesn't really matter because there's nothing in the territory that you're interested in. So this is a threshold model that does depend upon territory quality as an issue that you would be concerned about, because you're going to be residing in that territory and taking advantages of the resources in that territory. And this model makes the assumption that the first female on the scene gets the most resources, and any females that come after that get fewer resources. You could make this a polygyny threshold where you had the first female and the second female and the third female and the fourth female, and each of these would have a line parallel to these other lines that are just lower and lower on the graph, so that you could basically see, you know, when do you enter into polygyny, and do you enter into polygyny with only one other female or with multiple females, and what are the conditions under which you would have many females in the territory of one male rather than just one or two females in the territory of one male. But if you're like a prairie chicken, you're not using any resources that the male has anyway. You just come in, you get sex, you get fertilization, and you fly off and raise your kids. In which case, this model would not work for that. The females are going to the males for a totally different set of reasons.
Got it.
It feels like the building is going to fall down. Right. So your book talks about the pied flycatcher. And so one of the questions that the book asks is, why do pied flycatchers buy into polygyny? And they present the scenario, and so males generally abandon the first female to find a second female. So this is just how the system works. So it's a description. So males mate with the first female, and then they leave that female to find a second female. The males then abandon the second female to go and help the first female with parental care. So basically, male one, let's say Fred meets Judy, mates with her, and then flies away to find somebody else. We'll call her Susie. Then he leaves Susie and goes back to Judy and raises the children of Judy, and basically leaves Susie to fend for herself. Fred is a creepy dude. for a pied flycatcher, but actually is just normal for a pied flycatcher. The second female suffers a 40% reduction in chick survival compared to the first female because she has mated with that male, but she's not receiving any of the parental care from the male because the first female is receiving all of the parental care. So the question is, why does the second female buy into this? Why would she do this? Why would she suffer this 40% reduction in chick survival? Why not go find an unmated male and be the first female with that male? What's an obvious reason?
Maybe she doesn't know that there is another female. Maybe she thinks she's first.
Oh, maybe she doesn't know that the other female is there and she thinks she's first. What else?
Maybe because it's better than 0 matings if you're guaranteed at least 40%.
So maybe males are in really short supply, and so better to mate with a male and be the second female than to not mate with anybody at all. What else? We go back to the hypothesis that some of you struggled with on the exam, which is the sexy sun hypothesis. What is the sexy sun hypothesis? A surprisingly large number of you missed a question about this on the test.
Isn't it basically that like males that have good genes then like pass those
No.
good genes onto their no?
Do not confuse it with good genes.
Isn't it just that, like,
Because if you have good genes, what does that give you?
Does it give you better fitness for your offspring?
It gives you better fitness, but what kind of fitness? Is it a fitness from viability, or is it a fitness from a reproductive viability, or is it a reproductive fitness, or is it a viability fitness?
Well, couldn't it be both?
But often it isn't. Think about a peacock.
isn't
You guys know peacocks, right?
isn't it just that, like, there's some attractive feature about the mate, and so they just want their offspring to be attractive?
Yes, that's it. So the sexy sun hypothesis is you're choosing the male because of the male's, whatever this male's sexual advertisement is. So in the case of, are you guys familiar with peacocks?
I mean, just like their colorful stand thing.
They're colorful, and the male has this gigantic fan. How many of you have ever seen a peacock fly?
They don't, do they? I don't know. I've never
They
seen
do, but they really suck at flying. You will also remember the widow bird. Recall the widow bird. You can see the screen, correct?
It's, for me, it's like frozen in half.
Oh, is it?
It's like trying to minimize, but it got frozen.
Yeah.
Is it doing any better now?
No.
No.
Okay. Look up some video of a widow bird. Look up some video of a widow bird flying, and you're going to see that they don't fly very well. Peacocks are terrible. Yeah, I showed you some pictures in class. So widow birds don't fly very well. Peacocks really don't fly very well. And as a result, it makes these guys presumably not very good at surviving. And so the sexy sun hypothesis is that you have a badge or some feature of your morphology as a male that females find sexy, but actually tend to reduce your viability, reduce your ability to survive very well. And so the only thing that the females are really getting out of you when they mate with you is the fact that other females are going to find your sons attractive and mate with them also. Because your sons are not going to survive as well because they're going to be saddled with this marker that makes them attractive to females of their own species but reduces their viability.
But like, doesn't that fall under the umbrella of good genes because you're getting good genes for reproducing?
So you're getting good genes for reproduction, but you're not getting good genes for viability. So normally the good genes deal is about you're being chosen, a female chooses a male because the genes that are in that male are going to be good for their offspring, and that applies to both females and males. The sexy son hypothesis really only applies to the male offspring. And in almost all cases, these are genes that produce something in the male parent that they must overcome in order to be around to mate anyway.
Which
So
would
it's,
indicate that they have also like good genes for survival, right?
or it could just be that they lucked out. So this kind of dovetails with the Hobby's handicap principle, this idea that that the reason that I'm still around. So do you guys remember the handicap principle?
Yeah.
The Hobby's handicap principle basically says that the badge or whatever it is that you display that then females are choosing on is an advertisement of something essentially that has to do with viability. So for example, in red-winged blackbirds, the vibrancy of the red patch on the wing is correlated with resistance to parasites. And you don't want to mate with somebody who has a high parasite load because those parasites get in your nest and then they affect the viability of your offspring. And so in that case, that badge is a honest indicator of some aspect of your viability, in this case, parasite resistance, as opposed to a really long tail, which basically just decreases your ability to fly, which is also going to be implicated in your ability to escape predation. And so you can see with things like peacocks that the longer your tail is, actually the higher the probability is that you're going to be killed and eaten. And so it has a real effect. But females are willing to basically set aside that viability cost of having that particular display because the advantages of the display outweigh whatever viability costs there are associated with it. So when we think of the sexy son hypothesis, we usually think of a marker or a morphological feature in males that negatively affects their fitness due to viability, but positively affects their fitness with respect to future reproduction, as opposed to a marker under the handicap hypothesis where the vibrancy of the marker or the elaboration of the marker is actually correlated with something that improves your viability fitness. So it's increasing both reproductive fitness and viability fitness. Does that make sense?
Uh-huh.
So we teach this to you, and maybe we don't make as big of a point of it. But when we talk about studies where people actually study evolution, we can think of fitness, little w fitness, as having two components: survival to reproductive age, and reproduction once you get there. And so oftentimes, because it's difficult to measure fitness directly, we measure surrogates of fitness. So for example, we might measure survival as a metric. We saw this in a number of studies where people looked at the survival we looked at this in snakes, where Brody had these snakes that he got from the mothers. He marked them and put them out into the field, and then a year later he came back and collected them. And in that case, fitness was basically survival over that first year of life. He doesn't have any information on reproduction or anything else. The frog paper that you guys are reading for next Wednesday has actually fitness quantified in terms of both mating success and reproductive success. Not only how many copulations did you get with females, but then also how many tadpoles did you actually get out of those mating events. That's a much more direct measure of fitness. But in the case of the Brody snake thing, he was measuring an aspect of survival, which is a surrogate of fitness, because you can't survive to if you can't survive to reproduction, your fitness is going to be 0. In the frog paper, those frogs that are in that paper were already adults. They've already gone through this portion of fitness, and so now they're measuring reproduction altogether. Your book has referred to this back when they were talking about the sexy son hypothesis in the sexual selection chapter. They talk about this as viability selection versus fertility selection, I think is the term that they use. To separate one's ability to survive versus one's ability to reproduce. So, sexy son hypothesis. When you look at the sexy son hypothesis, there has to be a really high degree of heritability of whatever the mating status is of the parent, the male parent. And when it turns out the probability of polygyny for males in successive years is really low, so that's probably an upper limit on what the heritability of it is. So chances are that's not an adequate hypothesis. 2 of the hypotheses that you came up with is the deception hypothesis. And one of the lines of evidence for this is that males set up second nests really far from the first nest. Generally, it's at least 200 meters, and I forget what the book said. It's sometimes up to a couple of kilometers away from the first nest. And then the other question is unmated mates, unmated males may be hard to find. And when they experimentally manipulated this, they found that females chose unmated males as often as they chose mated males when they were given a choice. And so what they concluded is that there's not really any evidence to support the sexy son hypothesis. There's also not any support for the unmated males hypothesis. So pied flycatchers enter into polygyny because they don't actually realize that they're entering into polygyny.
Can you
And so, yep.
Can you re-clarify the justification about the sexy son hypothesis?
Oh yeah. So if you do a simulation study to see, okay, let's say that there's something that male pied flycatchers have, a color pattern or a long tail or whatever it is, that thing has to have pretty high heritability in order for that thing to be the object of selection and to account for why females would suffer a 40% reduction in chick survival. You would have to have a really high heritability of whatever that thing is that makes you sexy to other females in order to overcome that reproductive hit that the females take by being the second female. Now, if that thing is really heritable, it must be pretty important and pretty useful in males being able to achieve polygyny when they go to reproduce. Well, as it turns out, the probability of a male being polygynous, male offspring being polygynous when they reach maturity is really only 29%. And so whatever you're getting out of the parent out of the male parent that would make the female want to enter in polygyny has to result in the sons achieving polygyny easily. And as it turns out, achieving polygyny is actually quite difficult. And so this number of the probability of being polygynous doesn't match up with kind of the expectation that would be required for female parents to want to suffer that reduction in survival of offspring as a trade-off for getting this sexiness in their offspring. If the thing that you're choosing the male parent for really makes you that much sexier, then you should have a high probability of polygyny in your offspring. And the probability of polygyny is actually small, relatively small in comparison.
That makes sense.
So they figured out that it's actually just deception. Presumably, the reason that males find a female far away from the first nest is so that the female that they mate with second doesn't know about the first female. You may be thinking of human parallels, or maybe not. In dunnocks, once again, another bird. So much of this literature comes from birds because birds are easy to study in this way. In terms of dunnocks, female reproductive success varies across a wide variety of mating systems. So dunnocks can either engage in polyandry, in monogamy, or polygyny. Needless to say, female reproductive success is better when the female has multiple males. It's lower when it's just her and one male, and it's even lower when it's her and one male and a bunch of other females. Male reproductive success is actually just the opposite. They do best under polygyny, they do worse under monogamy, and they do the worst under polyandry because they are potentially caring for children that are not even their own. And so what determines whether this happens, which one of these happens in this system where they have the capacity to do all of these, is based on the competitive ability of the male, the sex ratio of the population, and territory characteristics. If you find yourself in a poor territory, what are you going to probably see happening? If territory quality is poor.
Monogamy?
Monogamy, or what would even be better?
Polyandry, maybe?
Polyandry would be better. You have a mother and at least two fathers caring for the offspring. So when territory quality is low, you might expect polyandry to dominate. When you have a skewed sex ratio, if the sex ratio is skewed such that you have way more males than females, what would you expect? What would you expect to be the dominant mating system in this species that is flexible?
Polygyny?
So you have a lot of males to each female.
Never mind. I thought there were a lot of females.
No, sorry, I made the males big. So let me do it this way. So the male to female ratio is, let's say it's 3-to-1. What would you expect?
Polyandry.
Yeah, you would expect polyandry. If it's flipped, one male to every 3 females, what would you expect?
Polygyny.
Polygyny, obviously. So all 3 of these things. So competitive ability. If the competitive ability of males is high, what would you expect to see? Males competing with males for access to females.
Polygyny?
Yeah, you would expect to see polygyny. So all 3 of these characteristics interact to determine if you have highly competitive males, but males are really in short supply, it may be that the competitive ability of a male isn't enough to make up for the skewed sex ratio, such that there are very few males. Sorry. If you have a very competitive male and the sex ratio is such that there are a lot of males to each female, your competitive ability might not be enough to overcome that, and you may go towards monogamy or polyandry. If, on the other hand, you have high competitive ability and there is a skewed sex ratio such that there are a lot of males relative to relatively few females, you may expect those males to guard those females, accrue as many females as possible. Wait, I said that wrong again. High competitive ability, very few males relative to females. The most competitive males get a bunch of females in their territory, and you might tend towards polygyny.
So is
So
monogamy,
What's that?
is monogamy just like kind of like a perfect world intermediate, or?
It's somewhere in the middle. So let's say what would be a good graphic to kind of illustrate this? If you could make a three-dimensional graph, and on one axis is territory quality, from poor territories to good territories. You have sex ratio, where we're going to do this, where this is percentage male, so that 1 is all males and 0 is all females. So this is this axis. And then you have a z-axis going through the middle here, where this z-axis is male competitive ability. From very competitive to, once again, not very competitive. In this three-dimensional box, if you have good competitive ability, but very few males relative to females, and intermediate territory quality, that would put you in a three-dimensional space that would probably tend towards polygyny. If you have good territories, it would push you even further towards polygyny in this three-dimensional box. If, on the other hand, you have a lot of males, and these males find themselves in an environment with relatively poor quality, and they're not very good at competing, you would find yourself in this realm of this three-dimensional box that is basically on the polyandry side of things. So in this three-dimensional box, there are regions where these three things interact, and where you find yourself in this box will determine on whether you're going to be towards polygyny, towards monogamy, or towards polyandry. But certainly polygyny and polyandry are at the extremes of this box, and monogamy is kind of at the center of this three-dimensional box. Is that a useful visualization? I wish I could draw three-dimensionally better.
Yeah, I think I understand.
Okay. So here's a figure from your textbook. This is the fitness of females under monogamy. This is the fitness of females under polygyny. This is the fitness of females under polyandry. So obviously, polyandry is the way to be from the female perspective, because you have higher fitness, because you have more parents engaging in parental care. And one of the things that will happen with the female is the female will mate with both males within sight of one another, so that basically they have both males have mated with the female, but they also see that somebody else has mated with them, and they basically are going to want to stick around because they want future mating opportunities with that female as well, because they're uncertain about the paternity of the offspring. And so she kind of uses mating as a coercive strategy to increase male input, because those offspring could be mine, could also not be mine, but on the chance that they are mine, I want to be there and provide parental care. This is especially important if you're in an environment where there are lots of males and very few females, because your chances of going off and finding another female to mate with is not very high. And so it gains you to stay with the female that you know you've mated with, rather than to try and find a female out there that doesn't exist. Polyandry is really uncommon in non-human animal systems, and it's really uncommon in human systems as well. I mentioned this in passing one day. There's a culture, if I recall correctly, that is up in the Andes, sorry, not the Andes, the Himalayas in Nepal. And their social structure requires the payment of a dowry. And females are rare enough that females command a really high dowry because they are the limiting sex in this particular culture. And as a result, any individual male's family doesn't have enough money to put up a dowry to acquire a bride. And so you will oftentimes find families forming coalitions where they get together and provide the dowry among them, and you'll have a female with multiple husbands because of a skewed sex ratio that makes females very valuable, drives up the price of dowries, and then requires males to make coalitions in order to be able to afford the dowry for a female. But that's the only example that I can think of in humans where polyandry is the norm. So at the end of this chapter, they talk about how you can approach thinking about mating systems or studying mating systems. And so if you look at broader phylogenetic comparisons, so comparing pheasants to hawks, there are differences in life history constraints that will determine kind of the type of mating system. If you have precocial offspring, which is the case with pheasants, that requires less parental care, which might drive mating systems in a particular direction, as opposed to altricial young, these young that are born without any feathers and need a lot of incubating and feeding and everything else. Altricial young would tend to have a mating system that would move more towards monogamy or something like polyandry, where you have enhanced parental care. You can also look closer to one another in terms of phylogenetic differences to look at, for example, species within a genus, where the different species may have differences in their ecology, the availability of food, the availability of breeding sites and things like this, to look at how those things influence mating system, which mating system comes about across these different species. And then for social conflicts, which is a lot of what we've talked about in the last chapter and somewhat in this chapter, you wouldn't look to those phylogenetic comparisons, you would look at differences among individuals within a given species to see how do these potential conflicts of interest play out within a particular mating system. And so you could look at mating systems from all of these different perspectives, but it will require different kinds of comparisons in order to study these different aspects. How do social conflicts influence mating systems? How do differences in ecology influence mating systems? How do differences in life history constraints influence mating systems? And that is mating systems. I'll try and generate a three-dimensional graph of how that whole Dunnett thing works if I have time over the weekend. Questions? Sex allocation is coming up next. It was supposed to be scheduled for today, but we're a day off schedule. So read the chapter on sex allocation. Sex allocation, I think, is going to go relatively quickly because it's just not that complicated. But the thing that I probably want you to pay most attention to is the theoretical ideas as to why sex ratios should be balanced. And then I want you to pay special attention to environmental sex determination, because that's an unusual topic and it's difficult to think about why, for example, temperature-dependent sex determination would have evolved as a way of determining sex in things like turtles and alligators. And so it's unusual, and it's also difficult to explain evolutionarily, and so you should put some thought into that. There is PowerPoint for sex allocation that I put on the Moodle site today so that you would have it available to you if we got to it, and we just didn't get to it. So we'll pick that up on Monday. On Wednesday, we have the Kroll and Hodel paper, which is about mating systems in poison dart frogs. And then on Friday, we might or might not talk about altruism there at the end. Let's see how we feel at that point. Questions? Haley, you look bored. No? Tired? Yeah. Okay. Are all of you as ready for this semester to be over with as I am?
Yeah.
Absolutely.
Absolutely. How are the rest of your classes going?
Become a lot less exciting.
Oh, I'm sorry.
Everything is culminating at once.
Yeah.
Oh, is it? Okay. Well, hopefully having things due this past week is giving you a little bit of a break, at least as far as this class is concerned. Except for Alexis. Alexis has discussion on Wednesday. So we'll handle discussion the same way. She has a PowerPoint and she'll present that to you guys, and I'll expect you guys to be conversational and unmuted that day. All right, I'll see you guys Monday. Have a good weekend. Try and get some rest and do something fun if you can.
Thanks.
Later.
See ya.
Thank you.
All right, see ya. Alexis, you sticking around?
Yep.
All right, show me your stuff.
I will. Let me see if I can. Let's see. How do I? Okay.
Share screen button, it's at the bottom in green.
But then there's like a ton of options.
Just share screen. Just share me your whole screen. And luckily, you don't have student grades on your screen, so nobody will see that.
Cancel. Hang on. What? So I click share screen and then it says desktop 1, whiteboard, blah, blah, blah.
How many desktops do you have?
I just have this one and the PowerPoint.
Oh, well, PowerPoint should be one that's choosable, but you can also just choose your desktop. Just choose desktop. That's
Okay,
easy.
hang on. Desktop. Okay, I'm choosing, well, let me just choose PowerPoint. Then it says open system preferences.
Oh, it's probably a sharing thing. Just share your main desktop.
Okay, so hang on a second. So it says allow the apps below to record the contents of your screen.
Yeah.
Then I just do this, and then I.
Come on, Alexis. You're supposed to be a digital native.
Well, it says you can choose to quit Zoom. It says Zoom will be able to record the contents of your screen. Won't be able to record the contents of your screen until it is quit.
It's because I'm recording, because I'm the owner of this. Okay, so send it to me via email, and I'll just open it on my computer.
Okay, I'm like, why can't I? It should
seems
be
like a backwards way of doing it, but
okay. You could just stop the recording, also.
oh, I could do that. Well, let me see. I can do that.